My name is Andy Hope from Rancho Biosciences, and it's my pleasure to welcome you today to join us for this exciting webinar from wherever in the world and whichever time zone you're joining us today to learn more about integrating single-cell transcriptomics data at scale for mechanistic drug discovery. We're going to discuss the importance of using high-quality data to produce meaningful insights which you can have full confidence in through creating robust workflows to deliver analysis-ready single-cell transcriptomic datasets at scale. We will then hear about applications of using bulk and single-cell datasets at scale to deliver new insights in mechanistic drug discovery. So first, it gives me great pleasure to introduce our two guest speakers. Manuela Salvucci from BenevolentAI. She's the lead bioinformatics data scientist there. She's been there just over 3 years and leads a team of a multidisciplinary team responsible for the target discovery process, including multimodal data integration at scale, model development, and software tools. And her team focuses on identifying promising drug targets by integrating complex biological and patient-level data sets. Manuela holds a PhD in bioinformatics and systems biology from University College Dublin. Although when you hear her speak, I think you'll agree she doesn't have an Irish accent. Dan Rosell is VP of Data Analytics at Rancho Biosciences. And since 2016, he's overseen the growth of our bioinformatics and data science teams, spearheading the transformative initiatives like Single Cell Data Science Consortium and the Spatial Innovation Initiative. Dan holds a PhD in cell and developmental biology from the University of California, Davis, and his teams regularly contribute to high-impact publications, driving the application of advanced analytics. Before we introduce Dan Rosell, I wanted to just sort of set the stage as to why we're sort of doing this. It was sometime in mid to late 2020 that we first began working with clients privately on single-cell data sets, helping to standardize and analyze them for them. But it became apparent that the universe of publicly available single-cell data sets is very large. So you've got one hand, you've got this vast data lake of single-cell data sets available that pharmaceutical companies can use to increase substantially the number of single-cell experiments their scientists can use. And best of all, they're freely available at no cost. However, they do come with some downsides. The challenges being that there's no common standards that are used to deposit these data sets in the public domain. It is difficult and time-consuming to be able to fully exploit these and create them in an analysis-ready format. And we were finding that clients were spending as much as 80% of their time on the data wrangling and standardization part versus 20% on the high-impact analysis part. So hence we set about doing this in a slightly different way under the guises of a pre-competitive consortium, which we launched at the beginning of 2022. And at that point, I'm going to introduce one of my colleagues, Dan Rosell, who is a co-conspirator in this single-cell data science consortium effort. Dan.
Great. Thanks a lot, Andy, and thanks to everyone for joining today. So I'm just going to give a little bit of an intro. It's set in the context of our pre-competitive consortium, but it mainly is lessons learned that we have generated over the years from how to generate these analysis-ready data sets to ensure that you have high-quality data that are analysis-ready and AI-ready for any sort of downstream applications. So the initial consortium was conceived as a pre-competitive effort, bringing together pharma and biotech clients in order to take advantage of these vast public repositories and really deliver value to their internal research programs. And this initialized 3 years ago in 2001, 2002. Andy, you can move to the next slide. Where we had our 4 core members that began this journey with us. During that year, we developed a catalog for them to be able to select data sets and developed a data model to be able to organize that data in a cohesive way together. And we delivered 116 data sets. In the subsequent year, we added a couple of additional members. We saw some shortcomings in the model itself, and we expanded into those to specifically address some additional nuances in donor-related treatments and sample treatment characteristics. And we also started developing some reference level atlases, and those I'll go into more detail later. As part of this expanded work, we delivered nearly 300 data sets in the second year. And that brings us to our current year, 2024, where we've added additional analyses, additional annotation techniques, and we're on track for delivering at least 500 data sets. Actually, we're now aiming for a little bit more. And right now, we're actually making plans for an additional phase, which is going to initialize at the beginning of next year, and this will be mark the start of SCDS2. Where we will sort of expand for capturing more data. There's plenty still available, and sort of take advantage of some recent advances in foundational models and AI that's begun over the last couple of years. So on the next slide, this is just kind of an enumeration of the successes of this consortium. So we've put together a system that can very reliably put together these, and by the numbers, we've delivered 90 million individual cells organized across 850 data sets. These are provided in a variety of different formats to enable easy analysis downstream. 100% of the cells are annotated with at least one cell type, most of them up to 3. And they really do cover a broad range of both diseases, tissues, and various therapeutic areas. But these were all built upon the initial concept. If you go to the next slide, that really kind of highlights the idea that the essential steps for building analysis-ready data sets completely hinges on a couple of foundational operations. This includes sort of the planning and the documentation of how you want to address this issue. This includes developing of the data model and all of the other periphery that go around that, identifying them, discovering the data sets, going through the actual process of cleaning and processing them such that they're consistent. And then in the next step here, we validate these for AI readiness and analysis readiness with a rigorous panel of quality control metrics. This includes inspecting both the metadata itself and also the corresponding data, having developed a number of different programmatic and automated scripts that check the readiness of these data sets. And based on this foundation, we believe that you're able to then take these off the shelf, perform exploratory data analysis, perform integration operations, and build AI models, really delivering the highest value to your operations based on this really coherent and thoughtful preparation process. The next slide just kind of highlights the key elements of that. So it includes, it's basically in preparation of having an AI or analysis ready data set, you have to make sure that it is organized, free from errors, and clean, transformed into an appropriate format, and provided in an interoperable format such that it can be accessible to any end use and also is amenable to external annotation using things such as ontologies and external identifiers. And throughout the process, we ensure that it's consistent. So we develop a plan, we develop a data dictionary and a data model to align our metadata to, through the coordinated use of automated tools and subject matter expertise, we're able to consistently clean this data, load it into these analysis-ready objects, and then ensure that it's delivered in this suitable format such that it can then be integrated into a variety of downstream applications. The next slide goes into quite a bit of detail of our specific process, and I'm not going to belabor the point too much, but this is really an interplay between these 3 different entities. So we have sort of the end analyst, these members highlighted in orange, which are responsible for sort of identifying the therapeutic areas they're interested in, identifying individual data sets that provide the most value to their end goals. And then sort of iterating on this process, they provide us with feedback so that we can always improve the process. Our curation team is responsible for internalizing the metadata. They will also reach out to individual authors, harvesting as much additional supplementary data from whatever sources are possible, such that that can be integrated all together, systematized, made consistent, and bundled together. Our bioinformatics team then finally takes the starting material of raw sequencing files, bundles together the metadata, builds it into a consistent process, and then finally interconverts this into a final format that is the most usable and flexibly immutable downstream. The next slide enumerates, so this is our approach to, because we do identify that there are many different ways that you will be wanting to use this, there are also many different computational biology opinions about how they want to analyze the data. We deliver our data sets in a variety of formats, so the same data is delivered in both a ScanPy H5 file, a Seurat RDS object, and then a number of flat files such as CSV and Excel formats. The entire provenance of the data set is tracked in a README file. A QC directory is provided that has a number of snapshots of QC metrics and plots throughout the process. And this is all delivered in a systematic way such that they can really easily be leveraged downstream. Then the final slide I'm just going to share with you today is internally, we've started developing these reference atlases. This was sort of a no-brainer that we now have all of these clean atlases pulled together. Many of them are enriched for particular types of diseases and therapeutic areas or tissue types. And it really saw increased value to be able to integrate these together to provide a unified representation of such areas. Using these tools, an analyst is able to even more easily understand the disease, understand the interplay of the specific types of cells that are applicable there, and also use it as a tool to annotate additional data sets coming downstream. And so that's just sort of the highlights of how Rancho prepares data sets as part of this consortium, hopefully a little bit of lessons learned there. And I'm going to hand it over to Manuela to talk a little bit more about how they're integrating data and making use of it at BenevolentAI. Thank you.
Thanks, Dan, and thanks to the organizer for the kind of introduction and the opportunity to present our work today. Next slide, please. So I'll just give you a brief introduction to the motivation behind the work we do at Benevolent, introduce a little bit what we do at Benevolent in general, and also how we have invested in reproducible data integration at scale to kind of power our end-to-end target identification. And then given the audience, I'll zoom in on how we discover novel biology via integrate at scale, patient-level bulk and transcriptomic data, and how we are also developing a gene regulatory network inference model from single-cell data. In the next slide, I'll just give you some brief introduction about Benevolent. So we were founded in 2014, so we are 10 years old this year. We got an office in London, and we also have a lab in Cambridge in the UK. We are a team of scientists and technologists, and we really work side by side in cross-functional teams. We have three major commercial activities. So we focus on knowledge exploration tools. We have our own drug pipeline. And we also have our own end-to-end drug discovery platform, which we use both for our internal pipelines and also in collaboration with partners. And the focus of it today will be on our end-to-end drug discovery platform. So in the next slide, I just want to introduce the motivation behind what we do. As we're all aware, under the traditional pharma model, it takes a lot of resources to bring a drug to the market, and the drug discovery and development process carries an extremely high failure rate. And one of the key reasons have been identified for these failures is really the lack of understanding of the underlying disease biology, which often translates in a poor choice of the therapeutic target in the first place. So for us, it's very important to understand the underlying molecular mechanism that underpins a disease in as much detail as possible so that we can make the best choice of target in the very early stages. One of our fundamental hypotheses is that the proper way to do this is by truly integrating all the available biomedical data as completely as possible. So as a company, we have invested in integrating at scale multimodal data, focusing on the right patient with the right data for the right tissue, for the right target, for the right commercial opportunity, within the right safety brackets, and finally targeted with the right drug. And the statistics that we're showing here on the right-hand side are just there to give you a flavor of what we mean by scale. And we will focus on this talk on the patient-level data. So in the next slide, in the next slide, we are kind of showing how so pivotal is the discovery of a novel mechanism, and how this relies on having a very solid and comprehensive data integration at scale in place. So our approach to bring in is to bring in as much data as possible and connecting it all together so that we can break down the silos between data types and diseases. Now, I won't go into the details on how we are actually building this data foundation. I just wanted to highlight that we have multiple sources that are complementary, and they span structured ontologies, unstructured sources that we parse and extract insights from using our NLP pipelines. And you see in the yellow box some of the numbers that we can extract. We also have experimental data. So this could be data from clinical trials or from our own experimental data that we produce in the labs. It could also be chemistry data. And a major focus of our data sources is really on patient-level data, and that includes both clinical data from biobanks, genetics, and obviously omics data, which then we have bespoke pipelines to derive insights from. And then today we'll take a closer look at some of the omics bespoke pipelines. So in the next slide, what we can see here on the left-hand side is a screenshot that is generated with one of our internal applications that we use to track and visualize data dependencies within our platform. And what we can appreciate here is that our data is truly integrated, and this integrated data then allows us to have a really multimodal approach and provide us with a proprietary integrated view of disease biology. Also, I'd like to highlight that we have invested in a robust and as reliable and repeatable process, so we can update and upkeep this data freshness. So what we're seeing here is not just a static picture, but rather we have data release at a very frequent cadence that keep the data. And then additionally, because our internal pipelines are disease agnostic, we have programs across multiple indications. So we have chosen not to cover a specific disease indication, but rather we have integrated data across the board. So we are able to use these effectively for diseases across multiple therapeutic areas, which lines up very well with the data provided by the Rancho Consortium. And then if we just focus on the patient-level transcriptomic data, we have a lot of data. And as we can see in this slide here, we really want to use multimodal data because we want our models to go beyond what has been reported in the literature to prioritize targets. And we really want to take advantage of the rich patient-level data that we have integrated at scale. So as we can see from the bottom, we have developed a mask language model that helps us achieve exactly this by basically having a more general purpose reasoner that can effectively incorporate the patient-level data. Now, because this is a language model, to be able to use the data needs to be in the form of sentences, and this is a process that we call textualization. And basically what we do is that we generate insights at scale from patient-level data, and then we take these numerical tables and we convert them into meaningful sentences, and then these meaningful sentences are added to the literature corpus, and the model will then be able to reason over them. And the reason behind this, we can actually see it if we look at the top right. We can see, look at here at the count distribution of the literature corpus. That's what we see in blue. So this is basically the number of times a given target has been mentioned in the literature. Now, a good portion of targets are mentioned a lot in the literature, and that's what we call frequent flyers or just famous and well-studied targets. Now, if we look at the distribution in orange and green from alternative modalities, now we see that these distributions look very different from the literature distribution in blue. And this is because they're not being referenced at the same rate in the literature. So for example, if you just look at the distribution in green from differential expression, you can see that it has a lot of greater coverage on less mentioned genes. And these are basically the genes that are considered off topic when you would write a paper about it. So if you go to the next slide, please, we can see that now we have built a data foundation, and this data foundation can actually support a mechanistic approach. And what we do when we do that is that we focus on pathways and physiological processes, especially those that highlight the transition from a healthy to a disease stage. Now we will see an example that we have a lot of data, and that allows us to learn across multiple diseases that share the same mechanistic pathophysiology, and also lends itself to uncovering direct evidence and novel insights. And as a proof of concept during the COVID pandemic, we were able to quickly identify and get FDA approval for baricitinib by repurposing this drug from rheumatoid arthritis. If you go to the next slide, please, we can then start to dive into our target ID platform. So our approach to target ID can actually be broken down into 4 main steps. So we have the discovery of novel biology, we have the target prediction piece itself, and then we had the post-target prediction assessment and experimental validation. So the step one, the discovery of novel biology, is really a key differentiator for us. We are very mechanistic driven, and we have really zoomed in this approach to tackle the complexity of disease. And this is because of what we discussed in an earlier slide, that half of the clinical failures occur due to a poor understanding of the disease biology. So we really focus on identifying the mechanistic drivers behind the disease, using clinical and molecular subtyping in line with a given disease area strategy, which results in clinically relevant strategies that in turn increase the probability of translation to patients. And basically, we can think of this process as effectively setting up the biological question that we then pose to the system in step two. So in step two, what we do is that we take the biological question from step one and we do target predictions. And this process involves running a fleet of models. We have generated multiple AI models, and we run them flexibly depending on the biological question, the disease indication, and also the data availability at hand. We have multiple models, and including a mask language model. And here, the key differentiator piece that we have is around reasoning over multimodal data and explainability. And then step three is effectively assessing the rank predicted list of targets that we get in step two. Whereas in step four, what we do is that we do experimental validation, some of which we can do ourselves in the lab in Cambridge. And we won't go through step 2 and 4 in this talk, but we're going to focus on step 1. So in the next slide, we are going to dive into the mechanism of discovery. And as I mentioned, we have bespoke pipelines that can leverage this data scale. And the focus right now will be on the bulk and single cell transcriptomic for the rest of the talk. So in the next slide, we just have a graphical abstract of how the transcriptomic pipelines are connected. So in terms of bulk transcriptomics, we have developed a proprietary workflow that relies on latent variable models. And this workflow extracts, annotates, and selects molecular signatures from which we can then identify targets. And similarly, in the single cell space, we leverage data made available by the Rancho Consortium, and we extract insights by running a workflow that we have developed on top of SC-Linker, which is a published workflow from Avi Regev lab from a couple of years ago in Nature Genetics. And what SC-Linker allows us to do is to extract the gene to cell type to disease relationships. And finally, we have also an additional proprietary workflow that allows us to map bulk and molecular signatures mechanism onto single cell types, and this helps us inform assay selection, which is then a key step in experimental validation and portfolio entry. And again, we can do all of this at scale, so we can capture all of these into molecular signature atlases. So if you go to the next slide, please. So as mentioned, we have really invested in patient-driven disease modeling at scale. And what we mean by that is that we have built a comprehensive atlas of disease mechanism molecular signatures from patient-derived gene expression profiles by applying the latent variable models linked to patient subgroups that I just discussed. So as I said, this is an atlas. So how comprehensive are we talking about? So we can cover more than 200 diseases with roughly 1,000 data sets. We have identified 17,000 molecular signatures, which map to 24,000 biological themes, which are basically high-level mechanisms which we could prioritize to pursue. So what can we use this pre-computed pan-disease molecular signature atlas for? So we can use it to corroborate insight from the literature and from other sources, for example, genetics. We can also advance our mechanistic understanding of disease biology and inform mechanism selection by identifying distinct and shared molecular signatures across multiple disease indications. And we can interpret disease-specific data sets from our internal pipelines or collaborators' pipelines in the context of other disease indications. Also, if we take head and neck cancer as an example, we can see that on the bottom box. We were able to use our proprietary patient stratification workflow applied to latent variable model-based signatures to find a novel prognostic biomarker, which was independent of established demographic and clinical pathological features. And if you go to the next slide, please, we can see that we made a similar investment in the single cell space by joining the Rancho Consortium. So we have built a comprehensive collection of pre-computed pan-disease insight from patient-derived human single cell expression profiles, so that we can establish a gene to cell type to disease relationship. And here we're using both patient-derived gene expression profiles from individual datasets curated by Rancho as a part of the single-cell consortium, as well as some custom atlases that we have built internally. And to derive this insight, we have implemented an SLinker-based approach, which really allows us to compute cell type programs, focus on the cell type to gene relationship in the context of a disease, and also disease program, which focus on a disease-gene relationship in the context of a cell type. We are also very familiar with the nice coverage of the data sets from the consortium. We know we have good disease tissue and cell type coverage, and we have been able to generate tens of millions of proprietary relationships by applying our implementation of SLinker at scale on top of the Rancho data sets. So now that we are here, how can we bring together inside a scale from bulk and single cell transcriptomic? So if you go to the next slide, we can just see here a flavor of how we can use multimodal bulk and single cell transcriptomic data to systematically identify targets. So what we have is that we build an end-to-end proprietary modeling approach that identifies targets in a nuanced context, so in the context of a disease, tissue, and cell type, that makes use of integrated at scale, pre-computed pan-disease insight from patient-level bulk and single cell transcriptomics. So this is a four-step approach where we start from the bulk transcriptomics data, so that's our yellow box. We identify molecular mechanisms from disease and tissue-relevant data sets. We project it in the context of pre-computed pan-disease molecular signatures we discussed earlier. We can then identify patient subpopulations as candidate responders to mechanism-modifying treatment via all the relevant clinical pathological metadata that are available, and we can then integrate these relationships back into the knowledge graph. We can then map the bulk-derived mechanism from bulk in yellow onto the single cell types in blue, so that we can inform assay selection. And we can then, in step 3, have a deep dive into the cell type gene disease relationships by the SLinker workflow applied to relevant data sets. And finally, in step 4, we can identify the targets by transcription patterns and upstream regulators workflows from cell type specific mechanisms via bulk and single cell patient level backing. So if we go to the next slide, we can look at a practical example of this. This is an example how we were able to identify a novel mechanism underpinning disease progression in systemic sclerosis. So as described in the previous slide, we first identify a biological theme from bulk transcriptomic data. So that's the equivalent of what we've seen in the yellow box. Of note, this mechanism have been reported in lupus and other autoimmune diseases, but they had never been linked before to systemic sclerosis. And then, as we can see from the heat map on the left-hand side, this molecular signature really stratifies and differentiates healthy patient from systemic sclerosis patient with either pulmonary fibrosis or pulmonary arterial hypertension. And next, we were able to map these bulk molecular signatures onto single cell data. And we can see from the UMA plot on the right-hand side that the bulk-derived signature is enriched in activating myofibroblasts and lipofibroblasts. And we can then deep dive into these cell types, and using marker gene, we can see that the lipofibroblasts, which are basically a lung-specific fibroblast subpopulation that are really involved in surfactant production, is actually overexpressed in control patients compared to patients with lung involvement, either from pulmonary fibrosis or systemic sclerosis. And on the other hand, the patients with a lung pathology have a higher expression of a marker for activating myofibroblasts. So this analysis, together with deep dive using trajectory analysis, which I'm not showing you here, led us to the conclusion that this pathophysiology of systemic sclerosis might actually be mediated by this mechanism identified through bulk analysis by the transition from lipofibroblasts to activated myofibroblasts identified through the single cell analysis. So if you go to the next slide, please, we can then move on to different application. So this is as we can see in the next slide, it's another workflow that we are developing, which is about inferring gene regulatory networks from single cell data. Now, the rationale behind this, developing this approach is that we know that transcription regulatory circuits underpin key decision in cellular behavior. There are numerous examples in the literature of processes that are really regulated by transcription factors and impact and transition from a healthy to a pathological state when this dysregulation is disrupted. So c-Myc is a classical example. c-Myc regulates the cell proliferation. Its dysregulation leads to cell growth and potentially tumorigenesis. TAL1 and FKB are also similar examples. Now, our think is that we can model the relationship between transcription factors and target genes. And if we can do that, this will markedly improve our mechanistic understanding of regulatory programs, which in turn would help us predict outcome of perturbation experiments. So we go to the next slide. We can see that we've been working on leveraging the single cell data from Rancho to infer gene regulatory networks. Now, these regulatory networks are inferred starting from co-expression of genes across the data set, from which we can then identify potential links between transcription factors and their targets. And now single cell data is a key data type and is really amenable to this kind of inference, to the fact that we have a very large number of observations per data set, as we have hundreds to thousands or even millions of cells. If you use atlases, and a good granularity in terms of cell type annotation and disease coverage that is provided by the consortium. So we are currently working on exploring the applicability of GRNs in drug discoveries, using, for example, target biomarker discovery and target list assessment as our initial use cases. And I'll finish off here. I'll hand it back to Andy.
Great. Thank you, Manuela. It's very insightful. It will generate some questions that we'll take a look in a minute. Before we get there, I just wanted to introduce the theme for a phase 2 for the single cell consortium. Clearly, given the vastly expanding numbers of data sets, and this is obviously a projection based on historically what we've seen over the last 3 years, we're not going to run out of single cell data sets that we can work on and that can be providing benefit to drug companies in an analysis ready format. So we've been discussing with the current membership of the consortium and with prospective new members, the establishment of a phase 2, where we will continue this, the work that you've learned a little bit about today. And our plan is to kick this off sometime in the late January timeframe. And we see 3 main areas for this work. We can deliver even more data sets. We'll expand approaches to cell type annotation, as well as adding potentially other data types. And this will allow us to build, significantly even bigger foundational single cell models. Everything we do in these efforts is prioritized by the members. And to give you some sense of the extent of the projections here, so when we finish single cell 1 at the end of February, we will have surpassed 100 million cells worth of deeply harmonized single cells. And this is a conservative projection, I feel, but we will at least double that by a follow-on phase for single cell 2. And we welcome the opportunity to discuss that with you and colleagues in your companies.